So I will have your labs back to you on our next lab day on Thursday, but looks good. Have you guys had fun with those? Those are some of the basic techniques that we're going to be using as we step through this course. So I want to make sure that we do a little recap on the difference between a dichotomous key and a phylogenetic tree, which were the two types of sort of systems of thinking about plants that we were trying to kind of model in our activities last week. Okay. So when we're talking about our dichotomous trees and our phylogenetic trees, the dichotomous keys you should have been using in this booklet on Thursday. So our book overall that you're looking at, booklet on trees in Minnesota. So we went to a specific section, the conifers, okay? And this bit on the first page, the sort of choose your own adventure piece, that is the dichotomous. Okay, so you'll find these in various types of field guides, manuals, things like that. This one you might take out into the woods if you were trying to identify the trees around campus or something like that, right? Minnesota trees would do well for you there. So they're going to be specific to some topic, some region, something like that. The key is this little outline. The rest of it, the pictures, that additional information is separate from the key itself. That's to help you check the key, help you confirm, but it's not part of a dichotomous key itself. So it may or may not be included in a manual. Realistically, probably. Okay, so dichotomous key is a series of what we call couplets. Couplet just being like two lines, right? So two choices. So the way you should have been stepping through them was looking at that 1A versus 1B, right? So you looked at the plant, you decided whether those leaves were scale-like or ball-shaped, or whether they were linear shaped. And if we didn't know what those terms meant, at the end of the course, there will be some terms that you're expected to know, but I didn't expect you to know anything last week. On the page previous, or the picture, some of those definitions of what they were talking about. So if you're looking at a dichotomous key and you don't know what the terms are, hopefully it defines them somewhere. If not, you look them up and ask someone. And so you make that decision, 1A versus 1B, decide yes, they're scale-like or ball shaped. You then go to 2A versus 2B. You decide no, they're linear shaped. You're then deciding 3A versus 3B, and you step out like that. So it's like a choose your own adventure book from when you're a kid, if those still exist. So in order to effectively identify plants using a dichotomous tree, a dichotomous key, you're going to need a specific number of couplets of these sort of either or statements. And it happens to be the case that you'll need n minus 1. So if you have 10 items to identify, 9 different couplets. Okay. So we do want to think about these and pay attention to these as we see different dichotomous keys throughout the labs in this course, because when you do your plant collection, I am going to ask you to create a dichotomous key for that collection. So you're going to put that at the front of your collection so that I can step through your little guide to identifying the plants you have collected, and I can find the right plant using that dichotomous key. So that's our dichotomous key. We're going to use it for identification, classification, if we find some new plant out in the field, out in the lab, something like that. This is different from our phylogenetic tree. So dichotomous key is just based on things you can observe. So it's only useful if you can look at it out in the field. If we tell you something about its evolutionary relationship, then ask you to assess that, and you are out in the woods, maybe you have a magnifying glass, maybe you have a couple chemicals, some basic tests you can do out in the field. You're not going to be able to sequence a genome out in the woods behind the school. So a dichotomous key has to be based on something you can observe, which may or may not mirror the phylogeny, which is the evolutionary relationship between the plants. So sometimes it can, that's great. But sometimes it doesn't, because we have more data other than just things we can observe with our eyes. So you're finding dichotomous keys in a thing called floras, which are just guides to different flower plants usually. manual, field guide, this is the kind of place you find them. Something's called a manual that usually indicates that it's going to have a lot more descriptive information included. Some of these other books may just be big, boring dichotomous keys. Fortunately, we don't have to mess with those too much in modern times. Depending where we are, we do have sometimes Wi-Fi on campus, right? So when we are practicing identifying outside, like I'm not going to pretend the internet doesn't exist. So we're going to use a combination of all of our different resources. One thing that's helpful is that our dichotomous keys like are limited, right? Like it's helpful sometimes that you're looking just at Minnesota trees. You're never going to get an answer for some tree that grows only in the Philippines, right? Or you might if you're using a bad app, right? So we're going to kind of consolidate some of our resources as we move forward. Okay, the resources are usually specific to a particular area and are not exhaustive lists of everything that exists, which is actually kind of helpful. Okay. So here were some of the images from that Minnesota Trees book. These were things you can use to confirm where you got to once you keyed out what you thought you had. So if you got yourself to a balsam fir, you could then confirm, okay, I had linear shaped leaves. Maybe I should measure how long they are, things like that. Confirm kind of on a checklist. Does this seem right? If not, that's fine. You can go back, you can try again, right? That's part of the process. We're kind of focused on process in this class. I don't really expect that anyone after you graduate is going to walk up to you and quiz you on did you memorize exactly what is in any one lab or not. So what I want you to be able to do is to think about things, to use these tools, and then to evaluate these tools and make sure you're using them correctly, that you're getting answers that make sense. That's kind of our goal. Okay. So a phylogenetic tree is a little different. So a phylogenetic tree can incorporate a lot more information. And we have a different goal when we're making a phylogenetic tree. You're usually going to find this in like journal articles, mainly, right? In a textbook. If we were using a textbook, you might find a phylogenetic tree or not, partially because there are arguments about phylogenetic trees. These do regularly get updated. And again, we're not really focused on memorizing everything that exists for the purposes of this class. We're thinking about systems. We're thinking about how do we create these systems? How do we use these systems? So a phylogenetic tree is going to be a diagram, and it's going to have branching points, right? It's going to have branching points where we have some type of evolutionary event, right? So we're trying to track descent. So what I have actually pulled out here are some of those conifers, those Minnesota trees, right? Several of them would be in the division Pinophyta, right? They are evolutionarily related to each other. So some of these, like the Pinaceae, were included in that Minnesota Trees book. I think we also had some Taxaceae in that Minnesota Trees book. You can see there are also a lot of other things that didn't appear in that Minnesota Trees book. So some of those dichotomous keys, some of those observable traits may follow along with a phylogenetic tree, but they don't have to. So as we're approaching each of these branching points, this would indicate some type of mutation event, some type of speciation event where we have a split. And then we have the descendants following through from each of those splits. That's what we're looking at when we look at these phylogenetic trees. Okay. So the one-page thing, we have a quick little phylogenetics activity to do. This one, constructing a phylogenetic tree. If you look through our introductory activity as I posted it online, this was on the last page, but since we had so much trouble with the projector and the printer, we didn't actually get to it. So I want you to take a second, and you can do these as a group. So what we're, or by yourself, either way, I don't care how you work. What you're looking at is a table of traits. So these traits are showing you ancestral traits. So those are like the older traits, so farther up on a tree, versus derived state. So a derived state or a derived character is something that appears after a mutation. So this is like the new mutation. Okay. And we're going to be looking at 5 traits, A through E, for these 6 species. So you want to go look at this trait matrix, look at these red circles that we're trying to define, and then we kind of want to identify what these species we get at the end are. do it myself now as well. There we have one of these. Thursday we practiced using dichotomous keys. This is a phylogenetic tree. So we're comparing, contrasting, internalizing these differences.
No, the one that. Oh, that one. Should have. Wait, really? Oh, so first. Oh, I didn't know that. No. So.
Anybody need a stickler at this point?
Oh, yeah.
Travel that far.
Thank you. Yeah. No. Yeah. Yeah. Yeah, I don't know. It's like so. Yeah, I wanted to. I mean, it was.
Okay. So hopefully this is not all that hard. But hopefully this does emphasize like what the difference is between a phylogenetic tree and a dichotomous key. So the dichotomous keys are what we were using as we were looking at those pine trees, those firs, those spruces, all of that. This is how we would use a phylogenetic tree or how we would construct a phylogenetic tree. So the basic process of constructing a phylogenetic tree is creating what we call a trait matrix. So this is the trait matrix. Matrix because it's like a box, right? We have data in each of these cells. And looking at this, we can use this for our purposes to identify, but realistically, if we were doing this as scientists, what we would have is trait data on each of these individuals, and we'd be back solving to try and figure out the evolutionary history, right? So this tree is the result of this data collection. Okay, so as you're looking at this, maybe hopefully you have a little bit of a targeted approach. So which of these branches was like the easiest to identify? Where did you start first? Yeah, hopefully we started down here at number 5, or we looked at number 5, we saw it was all the same here. All right, so down at the bottom, we can see that the species number 5 just has that ancestral trait, that minus for everything. So it hasn't collected any of these derived mutations, okay? So we just have it branching off. It is as it is, at least with respect to these mutations. So that's number 5. Okay, so then what are we going to tackle next? Easiest to pick the one that just has one mutation now, right? So we're going to look at the other side of this branch. So we now have a group here, all of which have some of our mutations. So looking over here, you see mutation D happens along the way to this splitting point. This top one over here is going to have only mutation D, a derived state only for that trait. So we're looking at our chart. We can see that number 3 has an ancestral trait for everything except D, where we see our derived trait. So what questions? I will tell you what I got for each of these, but before I just give you answers, questions about any of this that was tricky or anything that was confusing? And then I'll just confirm we all got the same answer. Okay. You can tell me if I did this too quickly, but what I got was 3, 1, 4, 2, 6, 5. Is that what everybody got? Any questions now? Okay, cool. All right, so that's what we're doing. A very, very simplified form of a phylogenetic tree. In reality, things get much more confusing. Things don't sort nicely this way. We don't have just individual traits that are varying at each of these points. Sometimes we have convergent evolution. So we have two things appearing at different places on a tree, but through separate evolutionary mechanisms. Sometimes we have traits that revert back to the ancestral trait after having evolved. So if you think, if you think about like whales or something, right? They evolved, developed legs, came on land, decided land sucks, went back to the ocean, right? So we have reversions in phylogeny. So it does get much more complicated than that. And we'll see a little bit of that when we take a look at those papers that I handed out. So this is the non-scary version of phylogenetics. Okay. So we're going to talk a little bit about classification and dive in deeper on this. But first, I want you guys to discuss a little bit about what is a plant. That was your homework, to think about what is a plant. So in each of your tables, I'll give you a whiteboard. I want you to let me know what you think a plant is. What are some sources you may or may not have consulted in order to kind of live out? Yeah, this is the combination. Specific cellular characteristics. No, I think or if you didn't consult any, let me know where you got the information in your it has any cellular characteristics.
We were all just. Yeah. That up and stuff, yeah. And I just. I'll just.
There you go, you're good. All right.
Yes. Every time on site. Regular arrow.
I'd also like you to discuss where you got this information, real quick. Okay, I'm gonna ask you to tell me some of these things.
Basically.
Talking to each other, so nobody feels singled out first. Okay, so I'm gonna start taking down some notes for us as a group. Why don't people just call out some answers? What are some things that you know about plants? Real fast, okay. So I got photosynthesis. What else do we know about plants?
Periodic.
Eukaryotic traits, yeah. Okay. What else do we know about plants? That it? I saw definitely more than 2 things on some of those whiteboards. Okay. Autotrophs. Cool. What else? Okay. Cool. So got some organelles involved. Some cell features. Oops, cell walls. Okay. Germination. Is that what I heard? Okay. Okay. Cool. Anybody want to add anything else to this list? Okay. So I'm not, oh, weird. Okay. So what about sources? What are some sources we were using for this? And it's okay if the answer is your 6th grade biology class or your 6th grade science class. 10th grade? 10th grade bio? Okay. Everybody use anything other than that? Okay. Google. Is that about it? Foundations. Okay, cool. Some previous coursework. Awesome. Okay. I'll accept that. So this is what we know about plants so far. That you heard about before. I'm not going to give you an exact answer for specifically what is a plant right this second. I'm going to save our whiteboard for later. I think it auto saves. There we go. We'll save it. Perfect. Okay. I'm going to share our screen again. Okay. Cool. So I expected some of you might have checked Wikipedia, but you didn't. So part of the reason that I brought this question to you at the end of the week last time is that what counts as plants is a little ambiguous depending on who you're talking to and exactly how specific they're being. So on Wikipedia, we have 4 different things that people are sometimes referring to when they talk about plants. Aristotle classified plants as like pretty much anything that was alive and wasn't moving, which is not one of our current definitions. Solid start, but me asleep, not a plant. Sometimes I feel like it, right? But some of these other definitions are still used kind of scientifically. So whenever you see this phrase, I don't know if you've encountered this elsewhere, but this is a Latin phrase, sensu lato, that means like kind of like loosely defined. Sensu stricto means like more strictly defined. And then I made it even more strict up here at the top. So plants broadly sometimes include green plants plus red algae plus some other algae. We say green plants, we're usually including green algae within it, as well as something called a stonewort. If you're looking on Google, people like to avoid some of this ambiguity, so they focus on land plants specifically, and sometimes specifically vascular land plants, because those are much more easy to define, to find a common set of characteristics that is really clear. And that is where we'll be focusing in this course. We'll be stepping through our land plants. So as we'll see, just because they're in this group of land plants, the embryophyta does not actually mean that they never live in water, right? So that's going to be a little bit of a tricky bit, right? So a little bit of that sort of returning to ancestral states can happen here. Okay. So we're going to think a little more about the history of plants before we take a look at those articles I had reviewed. So we're talking about classification and classification of plants. We're doing orderly arrangement into a hierarchical system, the one we use now. And so actually, I heard some of you say it previously, but they gave it to me as an answer, right? So for this class, when we talk about plants, we are talking about the kingdom Plantae, right? So we're going to be talking phylogenetically, right? Which is why I'm not going to give you an exact checklist of traits right now, because we're thinking about this evolutionary history. Okay. But that is not the only way that plants have been classified. So people interact with plants. It's really important for daily life, right? We eat plants. Super important to people across the globe pretty much forever. So plants have been classified by pretty much everyone forever. Earliest records we have of naming and classifying plants are about 4,000 odd years ago from India, but that has to do with when we find documentation from. So we have some beautiful pictures of documentation of early plants. Lots of places there's a particular focus on medicinal plants. So plants we can use as medicine, plants we can use to heal diseases, super helpful, in addition to food plants, knowing how they grow. Okay. And previous to our modern system of classification, where the scientific community has agreed that what we want to trace are these evolutionary relationships and build trees out of them, had a bunch of different ways of doing that. So an early way of classifying plants was through a form system. So this is a little more kind of like our dichotomous keys. It's an artificial classification, just grouping plants or grouping organisms based on characteristics that are convenient. So things you can see, right? So you might just look at a group of things, say like, okay, they have legs, how many legs do they have? So you might have been using something like a form system on our first day of class when we were identifying our different objects. We do use some of the concepts from these form systems today. So sometimes this is helpful to us, especially if we're thinking about like gardening, horticulture, growing plants, or if we're going through these manuals, these keys in the field, we'll often see them divided into, okay, are we talking about trees? Are we talking about shrubs? Are we talking about herbs, which have to do with the stem and the height and the sort of physical structure of a plant? We're deriving that from Greek people. I'm not going to ask you to remember all that much about names and dates because I think that's super important. So if there's anybody I think is particularly important for you to know, I'll try to highlight that for you, but this is kind of background of what we're doing basically. Another thing that we draw from some of these early systems are distinguishing whether plants are annual, biennials, or perennials. So you'll see this in garden sections when you're buying plants. Annuals really just grow once, then die back and they're done. Perennials are going to come back for you every year. So this is something that was incorporated in some early form systems. Some of these form systems were also used for describing medicinal plants and plant anatomy. So we have some important records from about 40 to 90 AD earlier that we still have their medicinal uses of plants. Some of them are cool. We may mention some of them as we go through different groups, like plants that have gone extinct that we still have records of. Some of them, disappearing to overuse. That's kind of cool. I like plant stories. I like stories about specific plants. Makes things fun. Easier to remember the names and dates. More worthwhile for me to remember the names and dates. Another thing we draw from these early systems are the concept of monocots and dicots. So we will be doing that in lab. This will become particularly relevant as we get into the angiosperms, the flowering plants, because this is something that is recapitulated basically by the phylogeny. So something that really is reappearing based on the evolutionary relationships, not just someone's observations about the physical form. It seems like it's actually kind of true about the evolutionary relationships as well. So we are going to start some seeds hopefully on Thursday. We'll see. As we watch them sprout and look at things, one of the first things that we'll see coming up is the cotyledon, this sort of like first little leaf. Monocots and dicots, most simply, are just defined as whether they have one little leaf that comes up first versus two. And so these are not their like real permanent leaves. These are these initial structures that sort of start off the photosynthetic process before a plant becomes really truly identifiable, having adult characteristics. Okay. And we'll talk a bit more about their traits later. We're going to skip that for now. Okay. So next, we had some sexual systems. Really, for the history of botany, this is the one guy people will really expect you to know who he was, Carl Linnaeus, because he's the guy who came up with our current method of naming genus species in Latin. He renamed himself in Latin. His name was like Carl, right? But he decided to be Carolus Linnaeus. to match his binomial system of naming plants. Okay, so that's part of why we remember who he is. For plants, systematics, plant biology, botany in general, he's also remembered because he collected a vast number of plants, and we still have his herbarium, which is the technical word for a collection of pressed plants. So we still have it, and we'll take a look at it later on. It's been digitized as well, so we can actually flip through this old herbarium to look at what kinds of plants he was collecting as he was developing the system, compare them to some of our modern plants. Both of them haven't changed that much, but distribution of plants has changed. We can look at some of these early kind of classification systems. So he was called the plant pornographer because he was really into classifying plants by their reproductive structures. So a lot of talking about plants focuses on their reproduction. Right? I don't think I will ever look at a fern the same way after learning about its spore process, its regenerative process. So we will be kind of dividing these plants this way ourselves, because this is one of the characteristics that does map pretty well onto the phylogeny. So he's classifying plants and animals by their sexual characteristics, by their reproduction, which was a little scandalous at the time. Okay. But it turns out flowers are an evolutionary sort of mutation. So this works pretty well for us. So we have a link here to his herbarium, which we'll look at later. So it still exists today. It's maintained by the Linnaean Society of London. Has 14,000 specimens and growing. Includes 4,000 type specimens. Does anybody know what a type specimen is? Okay. So this is something that is used pretty much across systematic phylogenetics. So I'm trained as a paleoanthropologist. So we use this too with humans, with animals. A type specimen is like if you find the first fossil, the first species, whatever it is, that one becomes the standard by which everything else is defined. So the first one is a record. So it doesn't really matter if it was exactly related to what you thought it was related to. If it has slightly different characteristics than you reported the first one that you find and give a name to, everything else kind of goes back to that. So this is probably my own little corner of the internet, but for a while, like Brontosaurus, that name disappeared. People decided it was no longer in use for certain dinosaurs because someone else had actually named that fossil species earlier, even though they didn't really know what they were looking at. I think it's back. I think they've decided that the Brontosaurus and the Allosaurus are different enough that they're different species. Anyway, the reason that is because the first one you find, guess the name, that's the type specimen. So it's like there's an actual kilogram somewhere, right, against which we measure all kilograms, right? Like our standard setting, a type specimen is the standard setting for a species. So we have these collections that have been digitized that people can use for research. We also have an herbarium of our own upstairs in a, I'm going to call it a filing cabinet, not going to call it a closet. I don't really know what to call it, a cabinet, I guess, just general cabinet, which we've begun to digitize. Pretty cool. I don't know if any of you looked at like the dates on some of those specimens that we were looking at last time, but we have plants collected around the area by the sisters starting in like the earliest one I happen to have seen up there is like 1911. So like 100 years. There are probably some older ones. I haven't looked at the date on every single one. They also were mailing specimens back and forth with people around the globe. So there's some in the Philippines. It's kind of cool. So we're in the process of digitizing our own collections because people can do research by measuring things from these photographs, looking at where they were found, looking at distributions of plants, how they have moved over the years, or how they have their ranges have been shrinking. And if there's botanical research you can do on these, basically. Okay. And we're going to move into our early natural system. It's kind of the next phase leading us to our modern system of plant classification, which involves sort of a combination of the sexual characteristics, also gross morphology. I think I mentioned it as an aside before, but does anybody remember what morphology means? This is a word I did not know till grad school, so it's fine. Okay, so morphology is just talking about like size and shape. Morphology, think about like animorphs. If you read those, right? Like it's like about the shape, what they look like, kind of. So morphology is saying like it has this many leaves, it has this many legs, it's this shape. That's morphology. Gross is another word for like big or like visually observable. Like technically, if you're looking at cadavers, that's gross anatomy as compared to like micro anatomy when you're looking at something with a microscope. So big visible morphology, big visible shapes. Okay. So overall, these early natural systems were based on physical similarities. Another term for that would be phonetic classification, because we're talking about like the phenotypes. So phenotypes are observable characteristics, as opposed genotypes are sort of genetic characteristics. They like to refer to as like natural affinities between groups of plants. Okay. Then biology had a big shift when Charles Darwin published The Origin of Species, and this is when we started to think about evolution. So we had a kind of a shift in the way we a big shift in the way we classified basically all organisms. So the post-Darwinian system is still a modification of this natural system, but what we're now trying to do is to classify plants and other organisms based on the evidence we have about what they're descended from. So we're trying to find their family, we're trying to find their relatives, we're trying to group things together based on our estimation of what happened in the past, right? We're trying to look at what we can see now and use that as a little bit of a time machine. This time machines are fun, but we haven't invented one yet. Sorry. So we're looking for ancestral relationships, and this is where we really develop this idea of the phylogeny, this tree of life where we're tracking descent. Okay. This is just a fun picture of some early sort of phylogenies, right? So our early phylogenetic trees don't look exactly the way some of our trees now do. Charles Bessey was an American botanist, and this is a pretty influential system, so I hear from other botanists. So we can see his little cactus because it has these little sprouts sprouting off of it. And these are some of his major evolutionary lines that he was able to figure out in like the 18 and early 1900s. So here we see those monocots arising again, even like those much earlier systems, but it's not monocot versus dicot based actually. Okay. Cool. So as we move forward in this course, especially through the labs, what we are going to be going through is kind of stepping through a modern plant phylogeny, specifically of land plants. So we are going to be focusing on what you guys think of as normal plants, right? That is going to be our definition for this course. And we'll just be kind of stepping through group by group. But I want you to take a look at what actual systematics articles look like. Systematic articles, at least in my field, have a reputation for being very boring and eyes glazed over and kind of math heavy. And I want you to know that after finishing a systematics course. So we'll talk a little bit about the data. At some point, we'll do a little bit of an activity in R to kind of practice for ourselves, understanding like how the math behind these things work, how the algorithms behind these things work. But I got you two very recent, I'm going to call these recent articles on phylogenetics of plants. We picked out green plants as our definition of plant here. And I recommend what I'd like you to look, because this is going to be very full of jargon. We're trying to get a sense of the field. The one in the Journal of Botany, that Flaxted phylogenomic analysis, has a really good summary of kind of like the history of these questions. So I would look at that one for kind of an explanation about what our questions are. Maybe skip a little more to the end for some of their conclusions. What I really like about the 1000 plant transcriptomes article is their figures are great. They have some awesome examples of phylogenetic trees and how these plants are related to each other. So I would like you to take some time going through those figures, trying to sort of prep yourselves for some of the topics that we're going to be discussing in this class. So, okay. Gave you some beginning reading questions for those, and I'm going to ask you to spend a little bit of time looking through these on your own, and then start discussing them as a group. These are pretty basic questions on how to start reading a paper. I may have some more specific ones for you as we start to discuss as a group.
I only have the top.
Okay. Everybody have both those papers?
You get the one.
Just update our titles here. And that's not my download, was just named. One more comment on reading scientific papers, right? We're not reading these to memorize, so I'm going to tell you my favorite thing to do when I'm reading scientific papers is to look for like science bites, right? If you ever go to a scientific conference, like what I like to do is collect gossip on who disagrees with who, right? And then watch those interactions. They're very tense and they're very amusing. And I find it's much easier to read papers if you think about the people writing them as characters who may or may not be having an argument with someone else, because papers really are advancing a scientific argument. And sometimes those scientific arguments also get a little personal. So as you're going through, I want you to be thinking about, like, when we're talking about open questions, right? Like, what's the kind of subtext that they may be referring to? Like, what seems like it might be something that people have been arguing about, and potentially asking really mean questions about it. I don't know how mean the plant people are, but the paleo ant people can get kind of mean. And I recently saw a big Twitter fight about T-Rexes. So I suspect the plant people are, have their own hot button issues. So I think it makes science more fun and more relatable. And it's true. So